Hi, everyone. Welcome to our second Delta Mosquito Vector Control District and Oxitec Joint Public Educational Webinar. Today's topic is our scientific publications, independent validation, and data transparency. How does that work? And we'd love to share that with you. Today we have two presenters, Dr. Kevin Gorman, our Chief Development Officer at Oxitec. Hi, Kevin.
Hey, hi, Rajiv.
So spare a thought for Kevin. It is 1 a.m. for him right now. So we're very thankful to have Kevin presenting with us today. My name is Rajiv Vaidyanathan. I'm the Director of U.S. Programs at Oxitec. Just a few intro before we actually jump into the meat of the webinar. These webinars are open to everyone, and please share. We would love to have more people join us. They are all recorded, and they will be available for you after the event, so they're all archived. And all questions relating to the webinar topic, you can just type in, we can hear them. We will answer them in batches if possible, but just please be aware that because our topic today really is on scientific publication and on data transparency, those will be the questions that we will answer first. So we will do our best to stay on topic. Please help us. And if time runs out, send us more questions at info@oxitec.com. All the questions and answers will be published in writing after the event with both external related online references. These webinars, this is just our second webinar as we're planning and hoping for a project in California. Our agenda today will be on scientific publications, independent validation, and data transparency. Kevin is going to cover the majority of these. And then a little bit about the community engagement and a chance to join the project. We want to make sure that you know that you can sign on online, and you can sign on when we visit you at home. We have been canvassing in Visalia, and so maybe we'll see you in person as well. And I'll cover some of that. At the end, we want to share. We want to spare some time for your questions. We've presented some of these slides before, and so I might just go through them rapidly as a context. A lot of people say, why now? Why are you in California? And we're here for one reason. Now, that is because of an invasive species of mosquito called Aedes aegypti. Aedes aegypti is globally an important vector of a number of viruses, including dengue, chikungunya, Zika, and yellow fever virus. Aedes aegypti has been in North America, mostly in the southeastern United States, for centuries. It was introduced into North America, but never really established west of the Mississippi. As of 2013, 2014, we started seeing established local populations of Aedes aegypti in California. In 2013, Aedes aegypti was detected in Fresno and Madera and San Mateo counties. Soon it spread, it stayed in those three counties, and it spread to Kern and Tulare, where we are now, Los Angeles and San Diego counties. It was then detected in Imperial and Orange counties, and now Aedes aegypti is found in over 20 counties in California. There are some inherent challenges to controlling Aedes aegypti. It feeds and breeds in and around people's homes, as I'm sure you've noticed. I'm sure the mosquito burden has increased where you live in California. Because it feeds and breeds in and around people's homes, especially during the day, it is difficult with traditional mosquito control that sprays before sunrise and after sunset to really target this day-biting mosquito. And it's also difficult to control all of the sites where female Aedes aegypti lay their eggs. Female Aedes aegypti lay their eggs in containers, and so not in vast expanses of water, but in a wheelbarrow in a bucket, in a child's toy, even in a drain spout. And it's difficult because these are areas that you don't always think of as mosquito habitats. And so they are often missed or overlooked, or sometimes they're just hidden, what we call cryptic harborages and cryptic sites for larvae. And finally, Aedes aegypti, especially in California, is resistant to a number of different insecticides, in particular pyrethroid insecticides, which are traditionally used for their control. For all of these reasons, the fact that Aedes aegypti is an invasive species that feeds and breeds in and around people's homes, making it difficult to control, and that it's resistant to a number of pesticides and is an important vector of devastating viruses, we are targeting this particular invasive species, which does not belong in North America and does not belong in California. About a year ago, 10 different districts, mosquito control districts in California, expressed interest in us actually coming here. They were interested in participating in pilot projects, but the practical aspect is there is no way we have the manpower to do all of that for California. So we really settled on a Goldilocks spot. There are a lot of Aedes aegypti in Los Angeles and Orange County, but it's operationally difficult to get around. And of course, Sacramento and Shasta are smaller. It's easier to navigate, but they don't have the same Aedes aegypti burden. And so we're really focused on the Central Valley, where it's small enough for us and logistically feasible for us to navigate. And also, there is a serious Aedes aegypti problem in the Central Valley. And so all of these mosquito control districts in California were in our initial EUP, Experimental Use Permit Amendment, to the U.S. Environmental Protection Agency, or the EPA. We then really narrowed this down to the Central Valley. And although some of these counties have had Aedes aegypti since 2013, 2014, some are a lot more recent. And so we are partnering with Delta Mosquito Vector Control District in Visalia, California, on a pilot project. I'm going to turn over to Kevin at this point, and he'll talk us through a lot of the resources and references that have brought us to this point. Right now in California, we're really just conducting surveillance. We're talking to people. There's a lot of community outreach, but we've been doing this for years in Florida and in Brazil. And Kevin, I'm going to mute myself and you can control the slides.
Okay, many thanks for the introduction, Rajiv. Hello, everybody. Yeah, we're going to talk to you today, as Rajiv has described, about the scientific publications, and we'll take them in that kind of order. We'll start with the publications. We'll talk a little bit about independent validation, primarily from regulatory authorities, but also from others. And then we'll move on to a bit of data transparency and talk a little bit about some of the work that we've done most recently in the US. As Rajiv quite rightly points out, we've been doing this for a long time. We came out of Oxford University back in 2002, so it's over 20 years, and our company is predominantly filled with scientists. It certainly was going through those development years. Lots of scientists working on these technologies and bringing them right the way through from the R&D, through the R&D pipeline, if you like, all the way through to the field. Just a really quick reminder, just one slide on the technology itself here. These are male self-limiting mosquitoes. We release males only. They carry 2 genes. It's a self-limiting gene which prevents any female offspring from surviving, and a marker gene so that they can be identified as well as their offspring can be identified from wild ones back in the laboratory. Our male mosquitoes are released. They go out and they meet with wild female mosquitoes, and of those offspring, as I mentioned, the females can't survive. The males do survive, and they carry those 2 genes on and pass that on through subsequent matings with other wild females. and add extra value for each of those males that's released. Few key features there on the left and the right hand side. It's very targeted, of course, because it's a mating based technology, so it can't really affect any other species directly. It's not got a toxin in there, it's non-allergenic, it's proven effective in the field all the way through to commercial approvals in Brazil, for example. It's self-limiting in the environment, bottom right hand side there, so it doesn't persist. Once we stop releasing our males, they're destined to die, and away they go. Within about 5 to 10 generations, they're completely gone, and there's no ecological footprint as such left, other than the fact that some of those Aedes aegypti, those pest wild Aedes aegypti, have been removed from the environment. Okay, so onto the publications then. The link at the top there, oxitec.com publications, that's where you can find almost all of our 100+ scientific peer-reviewed articles. Not all of them are there, but pretty much most of them are. Since about 2013, we've published almost exclusively in open source journals to make sure that everybody can access them and that nobody has to pay for them. So there are one or two that are, especially the early ones, that are a bit difficult to access these days, but pretty much all of them are there, and that's right across all our platforms. So that's not just public health and mosquitoes, that's agriculture too. So that's the place to go to. Go to our website, click on the links at the top there about our technology, and in that list you'll see the publications, and you can go down and you can just access them, the vast majority directly right from our website. Just a quick overview of the peer review method. Scientific publications or peer-reviewed journals are ones where experts have reviewed the scientific work independently and concluded that it's worthy of publication, and publication, being a stamp of approval, if you like, from the scientific community or from experts within that scientific community. So starting at the top there, once an article is submitted, it goes straight to the editor. The editor-in-chief normally of a journal would push that out to a subject editor, and that subject editor would then be a specialist, potentially, if let's imagine this was an Oxitec manuscript on an agricultural technology, maybe that editor would be an agricultural specialist. He would then pass that out to reviewers that he personally selects. He would select those reviewers according to their profiles, according to his networks, people who are experts right in that particular subject. So not just agriculture, but now we're talking about the particular insect, or the particular insect and its application, or the crop in question, for example. Reviewers are chosen because they're experts, but also because they've got an established record in the field. Once those reviewers are chosen, and it's usually a minimum of 2, but usually 2, 3, or even 4 of reviewers, they get the paper, they review it, and then they pass their comments back to the editor. The editor then decides whether it's an exception, it's an accept for the publication, it's rejected, or whether it goes back for further improvements. Now, most journals, it depends upon the quality of the journal, but somewhere between about 50 and 90% of manuscripts get rejected. The top journals will reject higher than 90% typically, the very top journals. The most common output, probably for those that that get accepted, is that they go back for revisions. A very small percentage get accepted without any revisions. Most of those that get accepted will go back for revisions, then back through the review process, re-reviewed, often by the same reviewers, and then through to the editor. Once approved by the reviewers and by the editor, they can go into production and then finally publication. It takes a little while to do that process, of course. Several months is the absolute minimum these days. And previously, historically, it used to be a lot longer, and you could be in the publication process for maybe a year. Peer reviewers, who are they? As I mentioned, they're experts in the field, but they've got to have a very credible, established scientific history. They're selected by the journals I mentioned, but a couple of things there. They shouldn't have competing interests. Of course, they have to declare those. If they do, they don't get paid. So there's no financial incentive here. And that's really a hallmark of the peer review system, is that these are all volunteers. They're not named, so the authors don't know who the reviewers are, and they can't contact them directly outside the channels of the journal. What's judged by those reviewers and by the editor, it's a few key items here. Most of them are listed, but novelty is one of the top on the list. It has to be new. It has to be research that's not published anywhere else. It has to be original research, and it obviously has to be appropriate for the journal's subject matter to appeal to the readers. The integrity of that scientific study is checked, so that is the design as well as the analysis, the statistics, if you like, on those data, and then also the replication that's in there. And then, of course, there's the language, there's the grammar, and all the rest of the technical editing that's done. Okay, so on to Oxitec's publications. As I mentioned, over 100 publications now, significantly over 100, right across the platform since 2002, when we first broke out of Oxford University. What we've done here is just split them into, I think, 4 different categories to cover. Categories that are relevant, and I'll just describe a few of the papers and how we've done that previously. So this is just showing that we've demonstrated our platform in multiple mosquito species. Mosquitoes, particularly Aedes aegypti, that's the one that we're releasing at the moment. That's the one that we first developed at Oxford University, and that's the one that we're looking to release in California and have been releasing in Florida. So 5 papers here, just showing you that we've got a good blend. We can get this technology to work in different mosquito species. These mosquito species are typically those that transmit diseases. So you'll see here Aedes albopictus, Aedes aegypti, both vectors of neglected tropical diseases, and that's Zika, chikungunya, dengue, yellow fever, and a few others. There's also bottom right there, you've got Anopheles stavensi, and this is a malaria vector. So this particular species is just one of many that vector malaria, and is a particular problem in Asia and more recently in the Horn of Africa. And then top right there, we've also got another publication. Sorry, in the middle there, we've got a publication in Culex. Transmitting viruses, again, animals, humans, and also a severe nuisance in a lot of places. So I'll just move on to the next slide. And you can see that, just a quick summary here, there's 4 different species here that we've proven this technology to work in. It's very applicable across these closely related species. Excellent, therefore, at preventing mosquito-borne diseases, not just those that are transported by Aedes aegypti or vectored by Aedes aegypti. There are other biological techniques that can do that. Sterile insect technique is one of them, for example. But what we have in this genetic technology is something that really doesn't impair the fitness of those mosquitoes. So those male mosquitoes are extremely fit. They carry a couple of extra genes, but they have no real cost benefit. There's no real cost of carrying those particular additional genes. They're not subject to an irradiation program, and so they are particularly fit, just as the wild ones would be. And because of that self-limiting genetic sexing that we use in the 5034 strain, i.e. the males surviving and the females being killed, we have those extra males that I mentioned earlier coming through in each of the subsequent generations to work for us. So for every male release, we get tens, if not hundreds, of mosquitoes working for us. Some other publications here really focused on the biosafety. So you'll find these on our website. Again, lots of publications relating to the biosafety of these kind of platform. Again, some here on in mosquitoes, but not all in mosquitoes. There's some in agricultural pests too. A range of publications here. This could be relating to niche replacement, for example. There's one in the middle there relating to a study done in Panama, where we looked at what happens when we take Aedes aegypti away to Aedes albopictus. In that particular area, it was a coexisting insect species, and we looked to monitor for the abundance to see if removing Aedes aegypti had any effect. It didn't. Over the course of a full season, it didn't have any effect on the abundance of essentially a neighboring or coexisting species. And that was one aspect that regulators really like to look at. There are others here looking into feeding studies, looking into the presence of tetracycline in the potential exposure of these insects to tetracycline, which is essentially the antibiotic group, which can be used to switch the self-limiting gene on and off. Insecticidal resistance is a real problem right across the insect control. It could be in public health, but also in agriculture and animal health too. And so we're very careful that all of our insects have a very susceptible profile in that they don't carry exotic resistance genes. There's several reasons for that. One is that we don't want to introduce resistance genes that could be selected for by the subsequent use of insecticides, of course. And also, it means that our insects, when they're fully susceptible, could be controlled by conventional methods quite easily. And so there's a paper there on the left-hand side just documenting some of the insecticide susceptibility profiles of our Aedes aegypti in India. And those different types of papers are often applied to our different products as well. So we might repeat the insecticide susceptibility research for different strains, for example. A little quick summary then of some of those biosafety papers. No effects on non-target organisms. That's an established fact for many of our products, and as I mentioned, it's a mating-based technology, so non-target organisms just really aren't affected. But also we're looking at invasive species. Typically, these disease-carrying mosquitoes are the ones that are invasive, so Stevensii and Anopheles, Aedes aegypti, Aedes albopictus, all invasive, and removing those from an environment really restores the health of the environment as much as takes away from it. So that's a particular thing to remember when you're thinking about these disease-carrying mosquitoes. No evidence of niche replacement, as I mentioned. Aedes albopictus and Aedes aegypti, that relationship was looked at. And then no long-term persistence of the self-limiting gene is another one. So the speed at which it dies out, 5 to 10 generations, as I mentioned before. And then the biological comparable to wild-type counterparts, and that's in terms of insecticide resistance, as I mentioned, but also in terms of fecundity, in terms of longevity, in terms of other life history parameters. Field performance, this is an area which obviously a lot of people are interested in. How does it do in the field, of course, and of course this is the area where the insects get out of the lab and start to generate a lot of attention. Lots of publications here, again, mainly across mosquitoes, but we've released our mosquitoes in Malaysia, in Panama, in Cayman Islands, obviously the US as well. We've done caged mating studies in Malaysia, in India, in other areas. There's a lot of work that we could publish potentially from the field, But novelty is something that can sometimes constrain that. One of the prerequisites for a good peer-reviewed publication is that it's novel. And when you've got 3-4 field trials already with one particular product, then not every journal wants to publish a 5th. It's already been done before. And so that's a constraint that we sometimes come into when we're trying to publish field data, for example, and we've already done 2, 3, or 4 field trials that have been published with a particular strain. Here's a quick list of some of those publications for field work. You can see, as I mentioned, a couple in Grand Cayman, Malaysia there, 3 different field trials published from Brazil, one from Panama, one from subsequent one in Brazil in 2018-19, should be coming soon. And then also we've got the Florida Keys project, which we also expect to come out soon with a publication. So there's quite a number there of publications that, again, you can access from our website and you can read them through. They have all the data, including the suppression figures, including the release rates, right the way through. And sometimes they were multi-year programs. They weren't just one season. Mating and dispersal is one item that is prevalent in those. It's an aspect of biosafety that we touched on in the previous group, but this often comes through in the field work because that's where it's tested. And more these days, operational viability is really key as we move into the world of products rather than a technology, and start to think about how these insects can be applied to generate real impact. Making sure that they're viable is absolutely essential to generating impact. Impact is the key word there. It's no good if these just stay in publications. We want to make sure that these products actually have real world benefit and get to those communities who really need this kind of technology. Agricultural pests, we've got a lot of publications here. Some are in resistance management, where we've demonstrated in combination with BT traded crops that we can prevent resistance developing by introgressing the susceptible genes that we've talked about, making sure that those susceptible genes are diluting out the resistance in the pest insects that are naturally occurring in those areas. It's a real bonus for this kind of technology, and one of the strong reasons why we've moved to this particular platform from the earlier Generation 1 platform. There are, moths here, there are flies, there are coleopterans, beetles. That's because this kind of technology, again, not only works very well in mosquitoes, but it works well across the insect world. There are certain insects that it doesn't really apply well to, and that's more about the biology of the insect, and maybe releasing lots of males, for example. If they could mechanically transmit a virus, for example, that wouldn't make sense because you'd be adding to the problem. But in terms of viability, we can pretty much get this to work in most insects, and it's really about the biology of the insect that determines whether it would make sense to have this kind of control strategy available. Quick summary here, just as we've seen it with the other ones. Fall armyworm is a major pest, particularly of corn, but also of other really important broadacre crops. It's one that's very advanced. We have a commercial registration in Brazil for our fall armyworm, and it's an insect that develops resistance really readily, not just to insecticides, but to traded crops too. And so this is one where resistance management really is a huge bonus. We can release our fall armyworm males, and they will keep resistance from developing resistance to the crop that those pest insects are on, as well as providing suppression. Fruit fly, a range of fruit fly here. We've done Mediterranean fruit fly, olive fly at the bottom there. Closely related species where we can have very, very clean crops very quickly. Pink bollworm, again, another big pest, this time in cotton. We've done releases with pink bollworm in the US many years ago, and diamondback moth in the US as well. Diamondback moth is a crop principally of brassicas, but other field crops too. Just on the right hand side there, we've worked with a lot of agencies and organizations and universities to do our work. We always partner almost invariably because we're typically working in different areas where the partners know best, but also because partners have a great deal of expertise that we can draw on, and becomes a mutual benefit to have these collaborative projects between us. Okay, that's peer-reviewed papers, or our peer-reviewed papers covered. And now I wanted to talk a little bit about independent review, and then we'll start with regulators. Regulators, of course, set the pathway that we must go through, the process that we must go through to get our products registered in a particular environment. It's super important, of course, that the regulators have all the information that they need. They look into the peer-reviewed literature, but they also can do their own studies. They can request studies from us, and they can request access to all sorts of data, not just by permission, but also legally. Lots of regulatory successes all around the world. Here's a map, if you like, of the majority of them, right the way through our public health and agricultural programs. Contained work, that's work in a laboratory. Contained field work, cage studies, as well as open field work. So that would be deployments in the open environment. You can see here, so I'll just point out some key ones. You've got the Cayman Islands, which was the first one where we actually released our mosquitoes out in the open environment back in 2010. You've got our USA pilots in Florida, 21 and 22, currently ongoing in the Florida Keys today. We've got commercial approval down here in Brazil for our 5034. That's our current strain that we're releasing in Florida and hoping to release in California, of course. And then there are others on the agricultural side, in places where GM, is usually quite difficult to work with, such as Greece, for example, and some very good technical reviews and opinions from Europe on our GM programs from France and the Netherlands. So I'll talk a little bit now about the EPA and the approval process that we went through for our 513A and our 5034, more importantly, through the Environmental Protection Agency in the US to get approval for our current field trials. The EPA receive a huge dossier of information from ourselves. You can see here just some of the tests and studies. It was a 14-month process for our 5034 review from the EPA. They do a full risk assessment, and they use different agencies to ensure that they gather opinion from others. There was a total of 25 commission studies contributing to that dossier, a total of 4,500 pages, split into 70 documents. So a huge body of work submitted by Oxitec to the EPA for them to look at and start to digest and review as part of the risk assessment process. In addition to that, and to all the studies that you can see in the dark blue band at the bottom there, they then reach out, look at the peer-reviewed literature, and make their own assessment in the end about all the different aspects that allow them to come up with a final conclusion as to whether there is risk. If there is risk, what that risk level looks like, and risk is a function of hazard and likelihood. If there's a hazard and it's likely to happen, then there's a risk. If there's a hazard but it's not going to happen, it can't happen, there's no risk. If there's no hazard, of course, then there's no risk. And so that's pretty much the principle that a risk assessment is done on. They look at the hazard and they look at the likelihood of that hazard happening. And the conclusions from EPA were very clear. They decided that there was no risk, no appreciable risk to humans, to the environment, to animals, from releasing our male mosquitoes, and as a consequence, they approved in 2020 us to release in the Florida Keys and in Harris County in Texas at the time, although we haven't followed through with that project. And then subsequently, just earlier this year, we got approval for an amendment which covered further studies in Florida and also a federal approval to release in California too. A really good confirmation of the safety of this particular product. The EPA is world-renowned. It's got a fantastic reputation as a regulatory authority throughout the world, and is seen on by many as really the gold standard for those kinds of regulatory approvals. So what happens then? We've got a federal approval here for our AUP now that we've just discussed, and how the EPA came to their conclusions. Then it goes to the state, and so in this case it went to Florida, and also it's in California with DPR now, but Florida have approved this amendment, and we are releasing in Florida Keys this year. So with the same amendment that that is being reviewed by DPR, that's Cal DPR at the moment for state approval in California. Then once those EUPs are completed in terms of state approvals, then we have local approvals. So they're the mosquito district themselves need to approve it as a project, and once that's done, the project can happen. Once the project's happened and the data's in, that data is, by definition, it's an experimental use permit data, and that data is contributes towards a final product registration at the federal level. So the EPA then gather that data and look at the data produced and decide if that is sufficient in terms of biosafety, performance, efficacy, if you like, to actually warrant granting a commercial approval. And there may be restrictions placed on that commercial approval, or it may be a national approval right the way across the US state. Just one thing to say at the bottom here, that central blue protocols, metrics, and analysis are predetermined by EPA. It's a really important point. So these field trials that we're doing with our mosquitoes, they're done and designed in conjunction with the regulators. They're designed to generate the data that the regulators want to see, and therefore the regulators dictate sometimes exactly how we must generate that data. It's not something that we can move and shift around, and we have to then play by those protocols, if you like, once we're out in the field, gather the data that we've been prescribed to produce, and then submit that to the regulators. National Biosafety approval in Brazil, a huge accolade for the product, commercial approval all over Brazil without restriction, and we're already now gone commercial, so that we're selling to customers in Brazil. This particular 55034 mosquito, over 20 million mosquitoes released so far, protecting people in Brazil right now as we speak, giving people huge benefit. And we're extremely proud of that, and really look forward to the day when we can do the same in the US and elsewhere. Lots of world-class partners and regulatory record summarized here, really putting this up to show you the partners and the collaborators that we've worked with, some esteemed ones without a doubt. CDC, Gorgas Institute, Rasmussen Research in the UK, the USDA in the US, of course, DEFRA in the UK, so many funders, but also agencies who have looked at this, looked at our products, looked at our data, and worked with us to progress them through the systems. Okay, hopefully that was useful. Hopefully that stimulated some questions for later on, and please do get those questions in. Those questions can be put in now through the chat, and we'll answer those at the end. But I'm going to pass over to Rajiv now to talk a little bit about some of the work that we've been doing in the US.
Thank you, Kevin. If you can hear me okay. A little bit about our work in Florida, because in one of Kevin's slides, he did show that our Florida project is ongoing. We started our mail releases in April of 2021, and we ceased in November of 2021. And this year we started again, first week of May, with the expectation of releasing our non-biting mails through the summer in the Florida Keys. And just a reminder, male mosquitoes do not bite, only female mosquitoes bite. Like, males don't even have the mouth parts that enable them to pierce and feed on blood. So a common question is, what were your results from Florida of last year? And some of that is ongoing, some of it is still under analysis, but we can give you some high level. It is not published right now, and we're still collecting data, and we've started the next project for this year. A couple of the highlights include that our self-limiting gene was effective in the field. Now, what does that mean? You know, when you say effective, remember, the presence of this gene in female larvae results in the death of female larvae. So when we collected eggs from the field, hatched them, and we looked for which ones carried the gene because they're fluorescent when we see them under a fluorescent scope, which ones are not. So we can tell from the overall number of over 20,000 fluorescent larvae, every single fluorescent larva emerged as an adult male. So that means that not a single female emerged from over 20,000 larvae that were collected. Good evidence right there that the females that carry the gene die as larvae. We also compared dose rates. So we would release X number of mosquitoes in one yard and another number in another yard. And that was a very initial assessment to see, okay, what numbers are we really looking at that would be operationally feasible, that might be helpful, say, for a mosquito control district or for a consumer, for a homeowner in their yard. So it was a chance for us, and as a necessary question, outstanding question for the EPA, what is your dose rate? Our males flew about 50 meters from the release site, so about 150 to 200 feet, which is completely in line with all the evidence from the last 100 years of Aedes aegypti. Aedes aegypti never has enough energetics to really fly very far. And so our males performed just like the local pest males in terms of flight radius. We also found that our dosing, that is the number of males that we released in Florida, were effective in over-flooding the number of invasive males. And so our males were competitive and actually mated with the local females, which is the next point. How do we know they actually mated? Because we collected eggs at all these sites. And the eggs, as I said, we brought into the lab, hatched, And then we looked at the total number of larvae. Then we looked under a red filter in the microscope and saw which ones were fluorescent. That is, which carried the fluorescent gene that was also conjugated to the female specific gene. So then you have a number, right? You know how many are fluorescent and you know what your total number is. So we know that our boys were out there not just flying, but they were finding mates. They were mating successfully, and among those that had the gene the next year, every single one emerged as an adult male. We also found, without necessarily looking for it, that the cups where we were collected. So that was good. What we wanted to do worked out. In addition, we also collected eggs and larvae from what are called cryptic sites. So we referenced earlier buckets, wheelbarrows, jet skis, any place where water collected, the clay saucers at the base of pots. We collected from essentially the natural and artificial containers where female Aedes aegypti lay their eggs. And we found fluorescent larvae in those sites. This was just a real cherry on the sundae, because yes, we were collecting from the cups we put out, but it also meant that mated females were laying their eggs in people's backyards, and that is a place where it's really difficult to attack this mosquito using traditional mosquito control. So this was great. It actually went into people's backyards, resulting in the death of female larvae. And just to reiterate, no females were released, first of all, and then anything that carried the gene emerged as a male. So not only did we not release any female mosquitoes, we also didn't create any female adult mosquitoes, and any female that carried the gene died as a larva. And this paper, or I should say this project, was reviewed by a very prestigious scientific journal by the name of Nature. You can see that here, and that should be available as well. We're very happy that we could announce quantitatively the results. It is not a peer-reviewed paper at this point because we're not done. So we have not submitted that. But this was evaluated by Nature, which found that our results were exciting, that we're on the right path, and it's really the reason why we are continuing this year in Florida. In parallel, we are waiting to hear from the California Department of Pesticide Regulation, or DPR. Just to remind you, first of all, we had to get the permit from the US EPA. So that's at a federal level. Then we had to get approval from the Florida Department of Agriculture and Consumer Services, FDACS, which we did, and that's why we're continuing in Florida this year. We also are waiting to hear back from the California DPR. Presently, we are close with Delta Mosquito and Vector Control District in Visalia. We don't pay them, they don't pay us. All we get from Delta is really good intel on where the mosquitoes are. We cannot do this without them, and we cannot do this without neighborhood and homeowner awareness. We need to know where the Aedes aegypti hotspots are, what the seasonality, the geographic distribution, and most critically, the need for Aedes aegypti control in Visalia. And that is the very valuable input we get from Delta MVCD. And currently, we are canvassing, knocking on doors, talking to hundreds and hundreds of homeowners in Visalia who have been uniformly gracious and supportive of Oxitec. They have been inviting us to collect mosquitoes on their property. On that note, how can you get involved? If you are listening to this webinar, we might assume that you live in California. Maybe you live in Tulare or Fresno County, or want to get involved in this. Well, the easiest way is to go online. Check out oxitec.com/California. You can find the information there. In addition, in Visalia proper, we are going door to door, and we are handing out door hangers. We're handing out flyers, and they have a URL code on them. You can scan that with your phone, and it'll take you to our website. Tell us where you are and how you'd like to be involved. Right now, we're just conducting baseline surveillance. What that means is we're putting out a little cup to collect eggs, or we're putting out a trap to collect adult mosquitoes. At this point, baseline surveillance is going to help us identify hotspots so that we can actually work with homeowners to put release boxes in their properties once we have the permit from the California DPR. So yes, please get involved. We're coming to the end of this webinar. This is our second webinar, and you can expect a third probably in about a month, and that'll be the results of our community engagement, your participation, and consultation with Delta MVCD. I'm going to transition now to some questions. I see that we've had a lot of questions coming in. Remember, we are going to focus on the questions that deal with publication, scientific reports, data transparency, and if you have questions to follow up on what we've done in Florida, we would be happy to. So just so you know, that will be our priority. If, however, we do not get to your question tonight, that's okay. You can email us at info@oxitec.com. Thank you so much. I'm going to jump out of this, and I'm going to take a look at our question list, which has been curated. Let's see. Oh, I should have had this open already. There we go. And let's start. Kevin, I think I've spoken enough. I'm going to open this up to you. Couple questions here. Can you publish the same paper in different journals? And then a follow-up, do your partners, I guess by partners we're talking about the Florida Keys Mosquito Control District or Delta MVCD, do your partners publish independent papers about Oxitec projects?
Okay, thanks, Rajiv. Good questions. First one is a no. You can't publish the same paper in different journals. It has to be original. For a peer-reviewed paper, you can publish, popular articles or opinion pieces, in different places. But for a peer-reviewed journal that goes through the peer-reviewed system, it has to be new. And it has to be novel. So as soon as you've published it, if you overlap the data set, it won't get published in the second journal. In terms of our partners publishing, most often you're committed to acknowledge everybody who's worked on a manuscript, whether on the practical work or on the manuscript itself. And so typically, even if it's a small contribution, it would go in the acknowledgements. Usually partners maybe take the lead authorship on our publications. That would mean that they write them. They maybe supervise the writing or the generation of the manuscript, and we would contribute the product and some of the technological advice. And so usually it's the first and the last authors that do most of the hard work, if you like, and often that is the partner. So we try as a company to ensure that the partners can do that if they wish, but typically, no matter who's worked on the manuscript, it tends to go in at least the acknowledgement if it's not in the author list.
Thank you, Kevin. I might take the next one. When are you going to publish the results of your Florida project? So we're not there. We're still collecting and analyzing from last year. We are working closely with the Florida Medical Entomology Lab and with Florida Keys Mosquito Control District, FKMCD. This year, CDC is going to help collect as well. It is still ongoing. And we've started phase 2 of the project in May. So it won't be this year, I'll tell you that. The results of the Florida project, we will submit as a more formal peer-reviewed manuscript sometime next year. However, it shouldn't have to wait that long, and that's why we share these minor milestones along the way in webinars. We've done, I think, maybe 15 webinars with the Florida Keys Mosquito Control District, 2 with Delta. We present at open conferences, so conferences like the American Mosquito Control Association, Wyoming, New Jersey, the Florida Mosquito Control Association, the Mosquito and Vector Control Association of California. And so we've been presenting our 2021 results in public forums for mosquito control technicians. We've also been presenting at webinars, and we have a speaker series where we've been presenting to the community. So while we aren't going to have a manuscript on the Florida work until the Florida project is done, in between, we have been providing updates publicly, including reports to FDACS, the Florida Department of Agriculture and Consumer Services, and to EPA. Let's get back. Are you, Kevin, you or I could take this. Are you going to control other invasive mosquito species in California? Someone is aware of the Asian tiger mosquito as well.
Well, it's a fairly prompt answer, I think, Rajiv. No is the answer. In the future, we could do. The Asian tiger mosquito, Aedes albopictus, is one that we have on our radar. It's one that we've shown our technology works in. We have, candidate strains. So it's certainly one that we can bring through the system, but at the moment, no. And then certainly, in terms of the project itself, they're targeting just control of Aedes aegypti. And so one for the list, but not for the immediate short-term future.
And on that point as well, a question: you mentioned a number of agricultural pests. Are you going to bring the same technology to controlling agricultural pests in California? And I would like to point out that we have about 2 minutes left.
Yes, we are a bit like albopictus, but maybe I'd like to think within a few years we could have several agricultural insects of relevance to the US ready for moving into field trials, potentially even more advanced than that. So yeah, agricultural pests are well on our radar. Big challenges for the world. Public health, disease, human disease, but also food security. And they're the kind of challenges that we really like.
Our final question tonight from the public. I'm not a scientist, but I want to know more about your technology. Where can I look for information that is more approachable? So I guess, we've been talking about peer-reviewed journals, which might not always be easy for people to read or even access. So where can people read about what we're doing in a more friendly manner?
I'd say the website. That's the place to go. There's a plethora of information on there. It could be the start with the Oxitec.com website. Of course, there are all the scientific publications there. Just click on the links at the top and you'll find your way around. But there's also all the news articles. There's, descriptions of the products, descriptions of the technology in far more simple terms, links to all sorts of external information, and lots of press releases as well, which really are very digestible. So I would definitely start with the website, oxitec.com.
Oxitec.com/California. That brings us to the hour. Thank you, Kevin. I know it's late in your time. It's only 6 p.m. here in California. So
welcome. Real pleasure.
Like to thank our audience who joined us today. Thank you for your questions. Thank you for sticking with us on a frankly very dry topic. So we appreciate that. And we will see you probably next month. Thanks so much.